So I'll tell you a little bit about our center before we dive into it, not to promote the center, but just to give you the context of the talk. So our mission, being in a medical school, is to understand how the microbiome impacts health and disease, and then to translate that understanding for better therapeutics and diagnostics. And you'll see why that's important to us in a bit. So as Russ said, we're fully accredited, both a CLIA accreditation and a CAP accreditation, which we're one of the very few centers with this type of accreditation for metagenomics and microbiome research. So we kind of paved the way, and that will be part of my talk. It's a painful endeavor, I'll tell you, if you're considering. So I think Rob did the work for me this morning. I don't have to convince you that if you do a literature search, more and more laboratories are conducting these studies with a clinical endpoint. And so being in genomics, I think you guys understand better than anyone that the regulatory agencies kind of lag behind the science. And so you get from bench to bedside, but you don't have the regulatory structure for that smooth transition. Right? And so what we're trying to do is kind of pave that way for metagenomics. So in addition to literature search, if you look at the number of companies that are popping up with a clinical endpoint, they're just ever expanding. And then, of course, as Rob mentioned this morning, we had the government buy-in with the announcement of the National Microbiome Initiative. So for those of you that aren't familiar with this, I just want to identify or define a few terms before because if you don't understand those, then I'll lose you. And so the microbiome is just the entire collection of bacteria, viruses, and fungi, microbes, in a that naturally occupy various niches or in a given environment. And so we want to identify who's there, right? And then metagenomics is the use of modern technology to study communities of microbes directly in their natural environment without the need for isolation and cultivation. So that really was the key to this field of study, right? So the next generation allows this study this field to progress. So again, just kind of set the context, whereas Rob was talking about WGS, I'm going to focus on 16S sequencing. And so if you're not familiar with that, it's amplicon sequencing. So you do PCR with a flanking barcode region. The 16S ribosomal gene is highly conserved, it's about 1,500 base pairs, and it contains 9 variable regions. And so it's these variable regions that allow us to identify the different members. So it's a paternity test for microbes, if you will, right? It allows us to classify at the genus level. And then again, it allows us to capitalize on the sequencing capacity of the NGS platform because we can include these barcodes in our PCR primers and then later tease out which samples associate which barcodes, right? Otherwise, this would be terribly expensive and laborious if we were sequencing one sample at a time. So we typically pull 400 samples in one MiSeq run. All right, so what do we get with the 16S data? We get who's there, what relative abundance, and then how samples relate to one another. And so you can see these different sample sites clustered together in a principal component analysis. So back to accreditation. Why would we want to suffer through the pain of accreditation? And I'll tell you, it's a very thankless job because as researchers, would you like for me to come into your lab and tell you how you're now going to do your science and how you're now going to record things, right? And so it's truly a thankless job. So, but I think this statement really kind of underscores that, and that is that if metagenomic analysis are ever to evolve into a mature clinical diagnostic, an appropriate accreditation program that ensures quality testing, and patient safety must be developed, right? So kind of the basic components of an accreditation program are we need some sort of reference material so that we can distribute to various laboratories, collect the data, and compare. And then we need best practices to kind of standardize that, right? And so up until last week, if you did a literature search for best practices in metagenomics and microbiome research, you would see one paper published back in 2012. That's kind of surprising, right? As fast as the field's advancing, there really wasn't much there. So the accreditation program process, you apply for a CLIA license and you'll get a certificate of registration. CLIA relies on third-party accreditation agencies to do the full accreditation, and so we chose CAP, which was the most appropriate for us, although kind of a strange mix because it's the College of American Pathologists, right? And so they're just now wrapping their mind around next generation sequencing, so now we're going to throw metagenomics at them. They're like, we're not sure what to do with you. So what it boils down to is you meet the basic criteria, which is you have a sufficient facility, you have a clinical lab director, your folks in the lab are trained, you submit your application with your fee, and they'll send you a 300 to 500 page document that you must answer line for line. So it takes on average about 2 years to do this. But I'll boil it down to you because if I went through that, you would shoot me before I was done. So the first would be proficiency testing. So that is validating your lab. You need to validate all of your instruments in the pipeline. Competency testing is essentially validating your personnel. And then whatever method or whatever test you're offering, you must validate that, and that's kind of what we'll talk about in this future slides. So what kind of measurements would we take in the validation process? CAP gives you those, and so you need to answer accuracy. So that would be the difference between observed and an expected value. So if you have a known sample, do I get that answer back? And in the case of 16S, we look at map genera or ID. You have to answer precision, which is reproducibility. So you need to look at that both intra-run and inter-run variance. So we measure that as concordance of operational taxonomic units. So that could be repeated pools. So if I had, remember I'm pooling 400 samples into one run, I could then repeat that, and then that would be my inter-run variance. Right. You could create a reference material, which is what we're going to kind of drill down to in a moment. So that could be a mock community or an environmental control. You need to look at analytical sensitivity, which is a fancy way of saying a limit of detection. Right? So we've empirically derived that to be greater than 1% abundance. And so one of the things I told you we could measure in 16S is relative abundance. So anything below 1%, we consider that noise. So we'll filter that out in our validation studies. We need to look at analytical specificity. So can I correctly identify an analyte in the presence of some adverse conditions? So if I had a mock community, which we'll talk about what that is, and have specific members in that mock community, do I get those answers, those IDs back, say if I have a low Q30 score? So here's the test method validation study design that we followed for our CAP accreditation. So again, we have to validate our facility. So we sequenced a pool, and then we sent that same pool off to the Human Genome Sequencing Center at Baylor, had them sequence it, and then we compared the data sets. We had to validate our sequencing pipeline. So that's a repeated run or the inter-run variability. We validated our technicians, so we pitted our technicians against each other. Who's better? Lab automation, so our pipeline's fully automated from extraction all the way through library prep. If you're going to do that, then you need to run the same samples through a manual pipeline and then compare those and see if there's any variance introduced by your automation. We have various methods of extraction. We typically use the mobile kits. So we looked at mobile power soil versus power mag. Is there any variance there? Low quality run QC. So anything in your QC metrics, so you get that screen back if you're running a MySeq, Q30, cluster density. So any of those that are suboptimal, you want to take that sequencing run, repeat it, and see if you get the same answer back. And then again, proficiency testing. So that would be previously sequenced samples that you'd run through your pipeline blinded to your technicians. So it would be treated like any other sample in the project. So we'll switch gears now and talk about reference material. So all of those measurements are going to require some type of reference material, something of a known value that you can compare to, right? So when we applied for CAP, we asked for a proficiency test, which is something that you would purchase. If you were doing NGS, they'll send you samples out that they have a database with the sequence. You send them back raw data, they compare it to it. Well, like I said, they were just wrapping their minds around NGS when we came to them with metagenomic samples. And so, of course, they didn't have one. So that puts the onus on the investigator. You have to come up with some sample to act as a proficiency test for your laboratory. So before we go into the reference materials, I think it's important to define it, because these characteristics of the definition are something that you want to carry through when you're designing your own reference material. And so that is that a reference material is a stable, abundant, and well-characterized specimen that is used to assess the quantitative or qualitative validity of a measurement process. So we'll come back to these as we start designing a reference material. Is it stable? Does it maintain its integrity over time if I store it at -80? Is it abundant? Am I going to run out of that sample? And then is it well-characterized? And so that's super important. All right, so before you set off to design your reference material, ask, what questions can I answer, or what questions do I need to answer? And you can kind of boil that down to anything that creates variability should be considered when you design your reference material, right? Because you want to try to control for that. So the first question here, and this is by no means an exhaustive list of questions, but it's just a start to kind of get you thinking. So does the measurement process exhibit some type of bias? Is there a systematic bias? And so we know that there's a differential between gram-positive and gram-negative in terms of extraction efficiency. So if we're designing a mock community, probably want to put gram-positive and gram-negative members in that mock community, right? So we can compare those two. So then you have a known ratio of gram-positive, gram-negatives. When you do your extraction, you can go back and measure that. How should the data sets be compared for future studies? So if I have a prospective study, so we start now, and then in 2020, I want to compare back to what we've processed today. Do I have a way of doing that? And so a reference material would offer that, and it should be cheap and easily accessible. Can we improve upon the process? So we know there are some pitfalls in processing these samples. Sample handling is a big one. Extraction is a big one. And so rather than use precious samples to improve upon that, we might use our reference material. This is the perfect sample. You know exactly what you should get and what you've historically gotten. Do I get better values? So if I put my members at low abundance versus high abundance, am I picking up these low abundance members in my mock community? as I change my protocol. And then how sensitive. So again, can I discriminate between genetically similar strains? So I might want to put two strains that have a close genetic relationship. Can I identify those two members? And then what are the limits of quantitation? So again, if you put low abundance, what is my lower limit of detection? When does it fall off? All right. So we can break the reference materials up into essentially three categories. So environmental samples is the first category. So just as the name indicates, there are samples from a given environment which may be suitable as a reference material. So if I was looking at soil samples, then I may get soil from a certain area and that serves as my reference material. Or if I'm stool samples, we have what's called a generous donor. So we have somebody in the laboratory that donates stool. That person would be our chairman. So and somebody, the last time I gave this seminar, somebody asked me, well, what about abundance of that stool? I said, he's our chairman. He's full of it. So can it be obtained in high enough abundance? Can it be homogenized to remove any variability? So as I aliquot this out, is there any variability? Obviously, if there is, it's not a good reference material, right? So if I send a lab to Russ and then I send one to Austin, if they analyze them and I'm giving them material that's not consistent, then we're setting ourselves up for failure. Is it stable to store over long periods of time? This, as I said before, this is kind of the Achilles tendon of these type of studies. So sample storage is a big issue, and that's something that we want to improve upon. And so if we use reference materials to do that, that's a good use of these samples. So the benefit is that the composition will closely match the sample of interest, obviously, because you're taking it from that sample. The drawbacks is it's difficult to characterize. So it's a very complex sample. How do I know what the members of that sample are, right? And then lack of reproducibility. So if I was taking a sample soil, that sample soil could change over time, right? So pH may change, or if I'm taking a stool sample, your diet changes. It's going to change over time. So the next type of reference material we might look at are in vitro models. And so that would be the use of a bioreactor to simulate or recapitulate some type of ecosystem. So you may take stool, put it in a bioreactor, and then cultivate that, right? And so the benefit of that is that it's controlled environmental conditions. You can control the community makeup, virtually unlimited supply because you're cultivating it, and it's relatively reproducible. So the drawbacks is unable to truly recapitulate the ecosystem. So it's not exactly the same as the sample of interest as you did with the environmental samples, and there is batch-to-batch variance, right? And then category 3 would be pure microbial isolates. And so these are pure strains that you would order from, say, ATCC, right? Or something that you have in the lab that you could grow up, right? And so we would use these to develop our mock community. So the benefit is that it is a pure isolate. You know exactly what it is. Hopefully you have genotype and the phenotype characterization of those. Should be unlimited supply. You go back to your freezer stock, grow it up. And it can be manipulated to mimic the ecosystem of interest to some degree. So the drawback is it's inherently artificial, right? There's no way we're going to recapitulate that environmental sample, right? So there's properties such as metabolites and in matrix in that sample that you just can't put back in. So this is an image that was given to me by Emma Allen Vercoe. And it kind of summarizes those last 3 slides. And I think it helps to visualize when you have it all together, the compromises that you're making, right? So on the very left-hand side would be the environmental sample. So in this case, we're taking it from the stool, and then we would homogenize that, put it in aliquots, and send it out to various laboratories, right? And so the benefit of that is it has the complex composition of the sample that you're looking at, sample of interest. The microbial ratios are true to nature, because that's where you're getting from. Realistic matrix. The drawbacks are that there's limited material. It's not easily defined, right? So we don't really know what all is in that sample. And then it may deteriorate over time in storage. And there may be a differential there. And so some microbes may degrade at a faster rate than others. And so that would change your downstream analysis. The next would be the in vitro model, the bioreactor. So you take some portion of your environmental sample, try to recapitulate that environment and grow them up in the bioreactor. So then you're approaching the complex composition of this. Plenty of material to work with. It's scalable for your system. And then some of the matrix components would be recapitulated in that sample. So again, you suffer for some of the things that you do with environmental samples, so it's not easily defined. It may deteriorate during storage. And then some of the components may be unculturable, and so those would fall out, right? So then moving further to the right, you could take pure isolates and mix them in the same bioreactor and develop some type of mock community, right? And so that would be defined and propagated. You have unlimited supply of microbes. You just order more strains from ATCC if you run out. Easily defined and characterized, and I use this loosely, and we'll talk about that in a moment when I talk about the ECHO experiment. You can recapitulate some of the matrix components, and then it can be tailored to whatever your project's needs. So depending on the complexity of your sample of interest, you may require a more complex reference material, or maybe you can get away with a simpler system. So the drawbacks are the microbial ratios may not reflect the source material, or it may lack the complexity, so we're getting away from the more complex. And then finally would be the pure strains or the mock communities. And so the benefit of that would be, again, unlimited supply of microbes, easily defined and characterized, and then it can be tailored to your project. The downside is you can't recapitulate the matrix and microbial ratios, and then lacks the complexity. And so then if you look at the bottom of this slide, it kind of boils it down to there's a trade-off between likeness to the sample of interest and reproducibility. And I would add in here characterization. So on the far left, very hard to characterize what's in that sample, whereas on the right, you know exactly what's in there because you put it in there. All right. Much like what I said about looking for best practices for these type of studies, if you look for reference materials for these studies, you would find that they're hard to come by as well. And so what most laboratories do will use a previously analyzed sample. So that's what we did in our accreditation process. There were reference materials made for the Human Microbiome Project. So our laboratory was involved in that, and we have some of those around the freezer, so we use those in our ECHO study. Emma's group designed some of these reference materials in the RoboGut, but these have been exhausted, so no longer available. You could purchase mock communities from Zymo, and so that we ran that as a standard in our ECHO test. And then ATCC is currently in the process of developing mock communities for purchase. But that's a pretty limited access to these materials. So most groups will design them on their own, which is fine for my lab if I design it, but it doesn't help the community as a whole, right? Because there's no comparison between the laboratories. And so what we would like to do, and the reason that we formed IMSA, was that we can get together as a community and kind of identify what should go into these reference materials, and then to develop a resource so that everyone can access that, and then take that data and put it into a database, and so you can compare back to that, right? So, reference material characterization. As I said earlier, one of the identifying characteristics on the right-hand side was easily characterized, right? And so we want to know a taxonomic classification, right? We should be able to get that from ATCC when we're ordering those. The sample source should be known and accessible. You should know the growth conditions if it's cultivable. Should be readily available reference sequence. You would think, duh, that no problem, right? So being in genomics, you expect that, especially in mammalian systems, right? You just go to NCBI and put in your accession number, and boom, you have a reference material. My background is in genomics, and I've been in metagenomics for 2 years, so I was quite naive when we designed the experiment that I'll tell you about in a bit. And it was eye-opening and frustrating. You should know the genome size, GC content. So remember I said that anything that has the potential to introduce variability should be considered when you're developing your reference material. So GC content for GC bias. Known copy number of target sequences. Again, duh, right? And so if 16S is our target sequence, we should know how many copies are in the genome so you can do the math later on. And then if you have functional information, great, right? So then we can make some educated guesses as to what's going on in that community if we have functional information. So I don't know if anyone's working on this, but have any of you used Ingenuity, IPA, back in the day for gene expression analysis? No. So if you put your AFI data into IPA, it would come back and tell you, oh, you should look at these disease states, or this is a hint as to what's going on. And so what I'd like to see is someday we can do that with metagenomic data, right? So we put in, okay, these are the microbes we see. Oh, this is what's going on in that system. So I think that would be hopefully some bioinformaticians working on that. Okay, so kind of switching gears then to just specifically talk about a mock community, and this kind of sets us up for the cool stuff, which is the echo. Right? And so most of that information in those previous slides was taken from a workshop that was done at NIST, specifically identifying reference materials. And so that was kind of a survey of what people do and what people think should happen. And I think what it all boils down to is you need a fit-for-purpose reference material. There's not a one-size-fits-all, because when we had a crowd of 200, 300 people and nobody could agree, then how do you compile that information with something usable? And I think what we found is that it has to be a fit-for-purpose, or some type of modular design where we have, say, all of the fecal, top 20 microbes in a stool sample, and that would be a block. And then you could add to it skin, right? So the bacterial, as I said earlier, the reference materials should include gram-positive and gram-negative, because we know there's a differential there for the extraction efficiency. They should be known and accessible. And then we should introduce some type of range, so low abundance to high abundance, so maybe a order of magnitude 100-fold. And then there should be some genetic diversity that should simulate your sample of interest. And then include some that are closely related so you can identify whether or not you're able to identify closely related genomes. All right. So now I'll switch gears away from kind of the compliance and the reference materials and get to the cool stuff, like I said, is the echo. So last summer, NIST put on a workshop to look at standards for microbiome measurements. There's a quote that I put early on in my talk. Another group that I'm involved with, which is the metagenomics research group at ABRF, I was talking with a guy and I said, we applied for a CAP accreditation and they have no reference material. We really ought to tackle this. He said, you got to talk to this guy, Scott Jackson at NIST. So I sent him an email and it had that statement and he said, that's our mantra, you got to get involved with this. And he said, what I want you to do is, we have this workshop and we've got Rob Knight speaking, we got Joe Handelsman from the White House speaking, we got Kurs Huttenhauer. It's like all the who's who of microbiome are going to be there. And we want you to talk about compliance. And I thought, oh, Jesus, are you kidding me? Like, how could you make that exciting and follow these guys up, right? So I said, no, I got to do something cool. And so the thought occurred to me that we needed these reference materials. And what better tool than the echo for precision, right, to assemble this? And so it kind of came out of my mouth, and Scott's like, cool, do that. And then the thought occurred, how am I going to get an echo? But luckily, Russ in Austin had been in my lab a few months earlier, I guess, to do a demo with the echo. And so when they packed the echo up and left, after I stopped crying, I thought, man, what can I do with that echo? So the freezer breaks. I'm like, how could the echo help me? And so as that came out of my mouth, I got in my car and headed home, and it's like, I better call Russ. So I gave him a call. So I want to echo what Michael said earlier. Labcyte is fantastic about coming through on these things. I'm like, Russ, I kind of promised this. You think you could get me an echo in Austin? Well, let me see what I can do. And so he called me back. He said, yeah, we're going to ship an echo to you. It's like, oh, how cool is that? I said, how long do we have it? He's like, a week. I said, okay, we can fit this into a week. The week included uncrating and recrating, so we had three days to do this. But we had Austin on our side, so that was pretty cool. So here's kind of what our study looked like. So we want to compare manual assembly of the mock community compared to the echo, right? So one of the things that we want to evaluate is extraction efficiency. So I thought, okay, I need a genomic DNA mock community, and then I need a whole cell mock community of the same bugs, right? Okay. So what are the variables? Genome size, concentration of the DNA once I extract it. What type of measurements are we going to need to make? Well, we need to take the concentration prior to transfer and then after transfer, right? Okay, we can do that. And then downstream, we're going to sequence and then maybe do some qPCR. Right, sounds like a good plan. Now, how about the whole cells? So what type of variables are we going to encounter here? Shape, size, growth media, growth conditions. Right? So we need to take the similar measurements, cell count prior to, cell count post transfer, and then same downstream measurements, right? So WGS and then qPCR. So one of the things that it wasn't Labcyte's first rodeo to ship an instrument. So they shipped a document and said, hey, fill this out, make sure you have all these questions answered, right? And I kind of saw why afterwards. I don't know what it costs to ship that instrument. I'm sure it's not cheap. So anyway, after filling out the list and coming up with our plan, this is what we sent back. So I assembled my team, and okay, here's the information we need. So we're going to have to measure these microbes prior to and after. How are we going to do that? Well, plate counting, CFU, plating and CFU counting, not fun. Hemocytometer, I think we can dig one of those up. We'll use that. Spectrophotometry, probably not good. So we called Labcyte. What do you guys recommend? And they came up with luminescence using Promega's back to tighter glow. So that was one way that we measured. After the fact, I found out that Logos Biosystems puts out a pretty good cell counter, that it's a reasonable cost point, I think 15 grand. Unfortunately, we didn't have that at our disposal, so we were a little bit handicapped here. And then quantifying DNA, sure, we can do that. We have a qubit, we got a nanodrop, we can do picogreen. But if you think about what you're transferring, if your downstream is qPCR sequencing, your sub-nanogram quantities, right? And so that's very difficult to do. So we're kind of stuck with qPCR, no problem. We got a Quant 7 in there, we can do it for sure. All right, so what do we need for that? All right, I need you guys to go find out copy number for 16S. And then we know we can use that as a calibrator, right? So I send them off, and they come back, and they have this list of potentials for our mock community. And then when it said copy number, this is where my naivete comes in. It would say 6 to 10, 7 to 12. Like, what? There's a copy number. There's a value that we have to use in the math that can't be a range, especially when you're talking about millions of molecules, right? It just kind of amplifies. So it turns out that the microbiologists aren't nearly as good as the genomics folks, right? Because they don't know what the copy number of these are in a lot of these bugs. And so you're kind of hamstrung there. But the echo had already shipped, and so we're going to push forward, right? We're going to do the best that we can. So we did the transfer. We did our library prep, which is the 16S that I showed you. How am I doing on time? Oh, okay. See, I got excited when the echo part came. So we took our amplicons and we ran them on the fragment analyzer, which is Agilent bioanalyzer on steroids. You run like 3 96-well plates at a time. Amplicon came out about the right size. So we moved forward to the 16S sequencing. We used Curtis Huttenhauer's Metaflan to analyze our mock community. And lo and behold, I didn't tell you what the members of our mock community were a priori, so that was probably by design. No, it's actually, we got the right answers, so in the right ratios when we went and looked at the data. So we were able to transfer bacterial cells with the echo to build this mock community. So we looked a little bit closer and followed our analysis that we use in the accreditation process. And we use a software called Atima, which was developed by the bioinformaticians in our group. And here's kind of some of the output. We looked at relative abundance. So we obviously ran technical replicates. On the left-hand side is actually cheating. That's the Zymo mock community that you can purchase. And so you see pretty reproducible abundances. And this is the mock community that we developed with the echo, right? Not quite as complex as the Zymo, but you see it's very reproducible in the ratios. So although we didn't know the exact number, we were close and it was reproducible. Here's just a look at read count. So our negative controls came back with no signal. We had a nice distribution across these samples, and then a spike in positive control, as it should be. Again, just kind of a look at taxa abundance, and it's highly reproducible. We compared the replicates and calculated FDR adjusted p-values, and so you see these are all ones, no difference between those. And then beta diversity, so these would be the replicates, so they're highly reproducible, and so you can imagine an auditor evaluating your data. And I go, okay, this is a pretty picture. I get that these are almost superimposable, very closely associated on this principal components analysis. But what are the values? I want to see some numbers. So then we plotted the distance matrix that Rob talked about this morning. And so you see that there is the smallest distance. This is in between the automated and the manual. I'm close to. So that's enough of the data. So in conclusion, measurement quality is a critical part of any experiment, particularly important to complex samples. So I think we would all agree that these type of metagenomic samples are quite complex, right? And then RM reference materials are used to add confidence to quantitative microbial measurements. And that the use of reference materials represents a logical approach to standardizing experimental procedures, right? And so that's the whole idea. Can we standardize it across the community? And reference materials can increase confidence in experimental design and output. And so if I come up with some complex experimental design and I run my reference materials through that, then I can evaluate it before I commit actual samples to it. So to be useful, a given reference material needs to be consistent, well characterized, widely available, and inexpensive. And so that was back to the definition of a reference material, right? And I think what we kind of boiled down from the consensus of that workshop is that the best fit is kind of a fit for purpose. You evaluate your test material and then construct your reference material to reflect that complexity. Oops. So some of the future challenges are those that we found in the ECHO project. So there's really not a good way or technology currently to quantify DNA at sub-nanogram quantities like that. So you can do it with qPCR, but you need to know the sequence and the copy number before you do that. And then also for whole cell references, how do you quantify that at those prior to and after, right? And so the instrument that I showed you, so they are starting to come out with technology that can do that. And then we need a set of best practices. Sample collection and handling, extraction methods, analysis parameters. So with that, I'll close. And again, I would like to thank Russ and Austin and all the Labcyte folks for shipping the ECHO and the support, and also for asking me to come speak today. So thanks for your attention.